A machine learned to detect cancer from breath samples with 86% accuracy. It hasn't made a single headline. That is the opening line you will hear. It sounds unreal, maybe too good, maybe too cinematic, maybe even fake until you understand what that sentence does and does not say. Let's start with the discovery. Imagine a sample of exhaled breath collected in a controlled tube. It is not magic, it is chemistry. Every exhale carries hundreds of compounds from food, environment, microbiome, and cellular metabolism. Some cancers alter those signatures. Machine learning models can learn subtle combinations of these signals that are hard to see by eye. Now the first tension, 86% accuracy is a single number from a specific experimental design, not a passport to instant deployment. If the model was tested on one hospital's population, one sample protocol, and one timeline, that number can look strong and still fail elsewhere. So what does how it works actually mean? Step 1. Capture. You need consistent breathing protocols, stable hardware response, and careful controls for humidity, tobacco exposure, time since last meal, and medication effects. Step 2. Transform. Sensor readings become digital signals. Raw signal streams are cleaned, normalized, and converted into features. You can think of this as making the breath readable for algorithms in the same way that images are standardized before medical image analysis. Step 3. Classification. A model learns from labeled examples, known cancer cases, and controls. It is not diagnosing by itself in the public imagination. It is computing the probability that the input signature matches patterns in training cohorts. Step 4. Validation. Here the story usually gets lost. Good teams report internal validation, cross-validation, and sensitivity specificity trade-offs. Better teams add independent external cohorts. Without that third step, 86% can be a museum display number, not a clinic-ready signal. Now move to a practical visual. Show a split screen. Left side, one controlled study group with high signal performance. Right side, a real-world diversity chart with smokers, non-smokers, chronic inflammation, and comorbidity overlap. The question becomes, does the model hold when biology is messy? That is the first reason why many of these systems are not deployed yet. In medicine, you are not optimizing for trendiness. You are optimizing for harm minimization. False positives are not just numbers. A false positive screening result may trigger invasive follow-up, anxiety, and repeated tests. A false negative can delay treatment. In both cases, a wrong prediction is not simply wrong information. It is a clinical consequence. So the second reason is regulatory and legal context. Medical AI software now sits under much stricter oversight. Agencies expect model documentation, drift monitoring, post-market surveillance, and clear intended use. If a developer says this detects cancer, but only supports a narrow, pre-screening context, that mismatch is a major compliance and trust failure. In the same way that one blood test is repeated and verified before treatment decisions, this technology needs layered confirmation. Not one model. Not one dataset. Multiple checks. Let's talk about why this is still important even before full deployment. First, cost and access. Breath-based screening, if proven, could be cheap, non-invasive, and fast. That matters in regions where imaging is expensive or where people skip care because current pathways are hard to access. Second, workflow. A low-friction test can move patients toward earlier clinical consultation. Not skip the clinic, but book an earlier clinic with stronger directional signal. Third, personalization. Different breath profiles across geography, diet patterns, pollution exposure, and smoking prevalence mean models must be validated by population context, not treated as one global model for all humans. Now we need the why nobody's talking about it layer. Not all silence is censorship. Sometimes silence means caution. Developers may be sitting on promising findings while they run larger trials. Hospitals may be reluctant to integrate tools without liability clarity. Investors may prefer to delay hype until prospective outcomes are replicated. Also, the media cycle punishes false narratives. A single headline with 1% can backfire if subsequent readers discover contradiction, replication gaps, or overextended claims. In high-stakes health content, a cautious team is often quieter than a startup pitch deck. For the final act, frame what it means. This is not a robot replacing oncology. It is probably a probabilistic sentinel. It says, based on your breath pattern, follow-up may be useful. That distinction matters for trust. A sentinel reduces missed cases when used right and avoids harm when paired with clinical judgment. That is different from declaring a diagnosis and ending the conversation. For patient-facing narration, here is the language you can use. A breath test could become one more signal, like blood pressure today, not a diagnosis by itself. For hospital-facing narration, the value is in screening sensitivity with managed specificity and transparent uncertainty communication. And for policy narration, the real battleground is governance and evidence standards, not headline math. Let's run one concrete scenario. Scenario 1. A pilot lab has 86% accuracy in a controlled arm, low confounder variance, clean sample handling. Scenario 2. Population rollout across mixed primary care clinics. Suddenly humidity, medication patterns, and prior lung disease introduce noise. The model drifts unless retrained and recalibrated. That is why external validation is the difference between a paper and a protocol. Scenario 3. Model output is used as a second opinion flag, triggers confirmatory checks, and gets reviewed monthly with outcome audits. Here you may see real clinical value. To keep retention, add a visual every 45 to 60 seconds. First the lab claim, then the feature pipeline, then the validation cliff, then the safety gate, then the patient story question. If you are asking, should I trust this? The answer is conditional. The science is serious. The deployment is not done. This is where most breakthrough stories fail, not in the algorithm alone, but in the gap between controlled success and real-world reliability. This is how the story should land. This may be one of the most important medical ideas in the next few years, and it may not become useful if we skip what actually makes medicine trustworthy. When the headlines are quiet, sometimes the lesson is not nothing is happening. It is something is happening, and it still has to prove itself in front of patients' lives. And the last line should sit clean. A machine can smell patterns in a breath and flag risk with impressive numbers. It changes medicine only when the evidence is broad, the risk model is honest, and the workflow is safe for real people. That is the promise. Quiet, careful, and still huge.